Hello everyone, welcome to my YouTube channel Physiology Learning. I am Dr. Ram. In today's topic, we are going to discuss about sensory receptors. So, in CNS lectures, we are going to start with the sensory system, then we are going to deal with the motor system, then finally the higher function. And today is the first lecture of the sensory system, that is the sensory receptors. So, coming to the learning objectives of this topic, we will be discussing about the types of sensory receptors and mechanism of transduction of sensory stimuli into nerve impulses. So, this stimuli how it is getting converted into nerve impulses. Then we will discuss in detail about the tactile receptors, which is very important, and we will be discussing about the thermoreceptors also. Now, coming to the types of sensory receptors, we have 5 basic types of sensory receptors. Various book deals with different classification, but this one is a very common classification. So, first sensory receptor is mechanoreceptors. Mechanoreceptors means they are basically going to detect the mechanical changes, that is mechanical stimuli like touch and the pressure. Mechanical stimuli, this receptor will sense the mechanical stimuli. Coming to the thermoreceptors, as the term thermo indicates, these are temperature sensing receptors. These are temperature sensing receptors. Then coming to this nociceptors. What is this nociceptors? The nociceptors are nothing but the pain receptors. The nociceptors are nothing but the pain receptors. And coming to the fourth group of receptors, which is called as electromagnetic receptors. These electromagnetic receptors detect the electromagnetic waves like the receptors in the eye, that is for vision. So, the best example for this electromagnetic is retina, where the rods and cones are involved in transmitting the electromagnetic signals into nerve signals, rods and cones. Then coming to this chemoreceptors, this chemoreceptors we have discussed in the respiratory system also, and they also detect the chemical sensations. So, chemo means chemical sensation. So, basically they are going to detect the smell and taste sensation also, smell and taste. Along with that, it is also going to detect the various chemical changes in the body like O2 changes, CO2 changes and osmolarity changes. All these receptors are classified under chemoreceptors. In today's discussion, we will be dealing with primarily the mechanoreceptors, and out of the mechanoreceptors, there is one group of receptors called as tactile receptors, which we will be discussing in detail. Then we will be discussing about the thermoreceptors. The rest of the receptors will be discussed in their respective chapters. So, let us get into the sensory receptor mechanism. First thing is, for all the impulses, there should be something called as transduction. What is transduction? Transduction is conversion of one particular impulse into another impulse. Here what we are trying to do is, we are converting the sensory stimuli into a nervous impulse. Only when it is converted into a nervous impulse, it can be sensed by the brain. So, it is activated with the help of a potential called as receptor potential. What is this receptor potential? Any stimuli, whenever they excite these receptors, what they are going to do is, they are going to change the membrane potential. They are going to change the membrane electrical potential, and when this potential reaches a threshold, what is going to get produced? Yes, action potential will get developed, that is our famous action potential will get developed, and these action potential can be carried on by the nerves to the central nervous system or the brain to perceive the particular sensation. So, this can happen in 4 mechanisms. One is the mechanical deformation. What happens in mechanical deformation? Let us see. Suppose if there is a receptor like this and it has some channels present inside the cell. So, whenever any impulse, for example, let us take a touch, the touch is happening, what is happening is, there will be some mechanical deformation in the receptor. This mechanical deformation, what it will do is, it can open up the channels which are present inside the cell and can cause the entry of calcium or any ions which can generate a potential. This local potential is also called as transient receptor potential. Whenever this potential reaches a particular threshold, what it is going to produce? It is going to produce an action potential. Whenever this action potential is produced, it can be transmitted to the nerve. Then coming to chemical application, some of the chemicals, they also have the property of opening up the ion channels. Then temperature changes, what does temperature changes do? They will alter the permeability of the membrane. Either they can increase the permeability of the membrane or reduce the permeability of the membrane. Then coming to the electromagnetic radiation, what they do is they are basically going to change the receptor membrane characteristics. Whenever this characteristic features are changed, there will be entry of ions or closure of the entry of ions. Depending upon this movement of ion change, the receptor is going to respond. So what all of them are doing is, all of them are trying to change the membrane potentials, and all of them are trying to open up an ion channel and create an action potential. So coming to the mechanoreceptors, the mechanoreceptors can be further divided into tactile receptors, proprioceptors and other mechanical receptors. Tactile receptors are very important, and there are several tactile receptors. Their number is numerous, but we will be discussing about the four most important tactile receptors, that is Meissner's corpuscles, Merkel cells, Pacinian corpuscles and Ruffini's ending. This is one of the favorite questions of all the examiners as well as the entrance exam questions. Coming to the second group of receptors, which is proprioceptors, they include the muscle spindle as well as the Golgi tendon organ. Then other mechanoreceptors are also present, like receptors of cochlea, which is involved in hearing, as well as the vestibular apparatus receptors, which detect some motion like acceleration, either linear or vertical, that is the vestibular receptors of the equilibrium, and baroreceptors. Baroreceptors we will discuss in detail in the cardiovascular system, wherein we will understand there is mechanical stretch of the vessels which is detected by these baroreceptors. So all of these receptors are going to detect the mechanical stimuli changes. So coming to the tactile receptors, this is the classical diagram given in most of the books, wherein the first one is the Meissner's corpuscle, the second one is the Merkel cells or the Merkel disc, then third one is Pacinian corpuscle, and the fourth one is Ruffini nerve endings. So we will be discussing all these tactile receptors under four headings, that is the receptor description, then their location, then their adaptation and the best stimuli to activate the particular receptor. So coming to the receptor description, we can just understand from the diagrams. We can see here the Meissner's corpuscles are encapsulated. Here we can see they are encapsulated. These are dendrites encapsulated in connective tissue, and there is one more receptor also which is also encapsulated. Can you see it here? They are also encapsulated. So these Pacinian corpuscles are also encapsulated by concentric layers of the connective tissue. It is like an onion peel. They are in concentric in nature, and they are also the encapsulated nerve endings. So both Meissner's as well as the Pacinian are encapsulated nerve endings, whereas the Merkel cells and the Ruffini, they are just the expanded dendritic endings. Here we can see just the dendritic endings are expanded. There is no encapsulation. Whereas in Ruffini also you can see it here, they are just the expanded dendritic endings. And we can see here the Merkel disc is on the superficial layer, which is the epidermis, whereas the Ruffini's ending is in the deep layer, that is the dermis. Then coming to the location from the diagram, we can easily say the first two M, that is the Meissner's as well as the Merkel's, are located superficially, and they are also called as superficial receptors. Whereas the next two, that is the Pacinian and the Ruffini, are located deeply, so they are called as deep receptors. And the two corpuscles name are rapidly adapting receptors. Remember it like that, the two corpuscles which are encapsulated, we saw them already together, that is the Meissner's corpuscles as well as the Pacinian corpuscles. We can see here clearly both of them are rapidly adapting, whereas the other two, that is the Merkel and the Ruffini, are slowly adapting receptors. Then coming to the function, which is most important, the best stimuli for a Meissner's is movement. That is, whenever any object is moving or we are touching a moving thing, then this receptor will get activated, that is the Meissner's receptor will get activated. Whereas coming to the activation of Merkel's, it will help in the touch and especially the points, edges and texture. So whenever any object is there, we are able to touch it in different places and understand the texture and shape of it. Where does it help? It helps in one beautiful place called as braille reading. What is braille reading? Usually the people who are blind, they have elevated regions of the letters wherein they can touch the edges and texture and feel the letters, and they will try to understand this. So this Merkel is the one which is involved in braille reading, and this Braille is a French educator wherein he has identified this kind of learning for the blind students. Until date it is in use, and it is such a wonderful finding done by the scientist. So the Pacinian corpuscles is going to respond to vibration because they are deep. The simple touch cannot activate them, so a vibration can activate them, these receptors. Then coming to the Ruffini sending, they are also very deep, so either a prolonged touch or a deep pressure can activate them. So all these points are very essential, and all the differentiation between these four is very important clinically as well as for the entrance and Viva questions. Now coming to the thermoreceptors means these are temperature receptors. So basically they are going to detect two different temperatures, one is the hot, another one is the cold. The cold receptors are activated by the fibers, that is the A delta and C fibers. These are just the nerve endings of the A delta and the C fibers. This fiber classification we will deal it in nerve muscle physiology, and they are especially activated between 10 to 40 degrees Celsius. Don't think that cold receptors means they will get activated only in extreme cold. Then coming to the hot receptors, these are the dendritic endings of the C fibers alone, and they get activated between the temperature of 30 to 49 degrees Celsius. And it is said that the cold sensitive spots are more than the hot sensitive spots. Then coming to one very special receptor which is called as TRPM8. What is this TRP? It is nothing but transient receptor potential, and M indicates the methanol. Sometimes whenever we eat methanol or the elements which contain the mint, we feel a cold sensation. What do? They activate this particular receptor, that TRPM8, which is a type of cold receptors. So because of this activation of this receptor, we feel the sensation of cold after eating menthol or any kind of mint products. Then coming to the nociceptors. Here why are we discussing the nociceptors in thermoreceptors? Whenever the temperature goes below 10 degrees Celsius or above 50 degrees Celsius, it is going to be a freezing cold or a burning hot. At that time, it is not the temperature receptors which are getting activated, it is the nociceptors which are going to get activated. So this is about the thermoreceptors. I hope it's clear. Thank you for listening. Stay tuned for the future CNS lectures. Thank you so much.